The process of production of sperms by the testes and ovum or egg by the ovary is called as gametogenesis. Once the child is given birth and then she reaches puberty, then only the next step of division will start. So until then it gets stopped and it gets arrested. Spermatogenesis, the production of the sperms by the testes, is called as the spermatogenesis. Hello everyone, a warm welcome to today's session on chapter 3 of 2nd PUC, that is human reproduction. I am Dr. Divya, biology faculty, Vidhyaashram Pre-University College, Mysuru, Temple of Excellence. So in the previous session of this chapter, we had discussed about the male and the female reproductive structure, wherein the male reproductive structure was the testis. We studied the structure of the testis, and the female reproductive structure, the uterus and its related parts, we had studied in detail. So now, since we know about the reproductive structure, it will be easy for us to understand how exactly the process of reproduction takes place, wherein in today's session we shall study about two important processes of gamete formation, which is called as the gametogenesis and spermatogenesis. So to start with today's session, let us first know what exactly is gametogenesis. So gametogenesis, the primary sex organs that are there. So when I say gametogenesis, it is the production of gametes. So what are the ones that produce the gametes here? In the case of male, it is the testis, which is the male reproductive structure that produces the gametes, and in the case of females, it is the ovary, that is the female reproductive structure which produces the gametes. Of course, they are a part of the reproductive structure, but they are the primary reproductive structure because they are the ones that actually produce the gametes. So the primary sex organs such as the testis in the case of males and the ovaries in the case of females, they produce the gametes. So here when I say gametes, the testis will produce the sperms and the ovary will produce the ovum, or it is also called as the egg. And by that particular process is called as gametogenesis. So what is gametogenesis? The process of production of sperms by the testis and ovum or egg by the ovary is called as gametogenesis. So moving further, let us study about spermatogenesis. So spermatogenesis means genesis is to generate something. So what is getting generated here? Spermatogenesis, right? So the sperms are getting generated. So we shall look into how exactly are the sperms formed. So that we'll study under spermatogenesis. So in testis, the immature male germ cells that are present. Now you need to recall what are these germ cells. So germ cells are also called as spermatogonia. If you have forgotten, please go in for the last session of this chapter, that is the previous session of this chapter, and have a look into the video. You'll understand, because so there I've completely explained what spermatogonia is. So to brush up again, let me tell you the male germ cells, where is it present? So we all know that the testis is present inside the scrotum, right? So inside the testis, when you take a cross section of the testis or when you look into the testis, it is made up of 250 different compartments, right? Which is called as the seminiferous tubules. And in that seminiferous tubules is presence of the male germ cells, that is the spermatogonia. And these male germ cells later on, they undergo divisions to produce the sperms. So that we'll study under spermatogenesis in the coming slides. So in the testis, the immature male germ cells, they produce sperms by spermatogenesis. And this production of sperm actually begins in the male at the puberty. Of course, males, they will have their testis right when they are inside the mother's womb. At certain month, they will start to develop their testis. But at that time, the testis wouldn't be producing any sperms. It is only when the male reaches the age of puberty, it is at that time the sperm production will start from the testis. So the spermatogonia, which is present inside the wall of the seminiferous tubule, which is present inside the wall of the seminiferous tubules, it multiplies by mitotic division. Always remember, first division that occurs either in spermatogenesis or oogenesis is mitosis. Later on, they undergo reduction division. So that you need to keep in your mind, because this particular schematic representation that I have shown here, it is very, very important question in terms of examination. So that is why this is a very important topic. So where is the spermatogonia present? In the seminiferous tubules. As I told you, there are 250 lobules or compartments that are present, right, which are called as the seminiferous tubules, and in these compartments actually the spermatogonia are present. So what are spermatogonia? They are nothing but the male germ cells. So if those male germ cells have to mature, then first they need to multiply, they need to increase their number. So when they increase their number means, how will they increase the number? The cells need to divide, right? So when the cells dividing is nothing but it is mitotic division. So therefore by mitotic division, which is equational division, they will increase in number. So each spermatogonium is now diploid. Now can you tell me why is it diploid? It is diploid because it undergoes mitosis. Mitosis means it is equational division, right? So that is why. So it is diploid in nature and it contains, so since it is diploid, it will contain 46 chromosome or it will contain 23 pairs of chromosome. So this is about the first step in spermatogenesis. As you can see, once the male reaches puberty, his seminiferous tubules will start to become mature. So when the seminiferous tubules start to become mature means the cells that are there in the seminiferous tubules, they tend to divide. How do they divide? That is, the spermatogonia tend to divide. So the spermatogonia divides by what division? Which is mitosis. Mitosis is equational division. So therefore here they will be haploid in nature, and the primary spermatocytes, that is after mitosis, they will be haploid in nature and they are mature now. So they are mature. Because they are haploid and because they have undergone mitotic division, they will have 46 chromosomes. So this is how the first step of division will take place. So next, moving further, what happens we shall look into. So some of the spermatogonia that are there, which are called as the primary spermatocytes, they'll undergo again a round of division. Now here these primary spermatocytes will undergo reduction division, which is meiosis. Therefore, their chromosome number will be half. They will be haploid in nature. Can you see here? Some of the primary spermatocytes, what happened? They underwent the first meiotic division to produce haploid primary spermatocytes, which are having 23 chromosomes, because it was meiosis, right? Always I told you, first division is mitosis, then comes meiosis, wherein the chromosome number will be reduced to half, because we all know that be it the egg or the ovum in the female or be it the sperm in the male, both are haploid in nature. Only after they fuse and form a zygote, they'll become diploid in nature, because n plus n will eventually result in 2n, right? So that is why here, when the sperms are formed, they should always have half a set of chromosomes. So therefore, first division will be mitosis. After mitosis, it will undergo meiotic division. So that is what happens here. Now, a primary spermatocyte, it will complete its first meiotic division, which is also called as reduction division, because it is reducing the number of chromosomes there, and later on it will lead to the formation of two equal haploid cells, which are called as secondary spermatocytes. And these secondary spermatocytes, because they have undergone meiosis, that is reduction division, the chromosome number from 46 will get reduced to half. That is 46 divided by 2, it will be 23. Therefore, having only 23 chromosomes each. That is what happens. So, in secondary spermatocytes also, what happens? 23 chromosomes. Now, these secondary spermatocytes, again they will undergo a reduction division. So, the secondary spermatocytes that are there, they will undergo a second round of meiotic division and they will produce 4 equal haploid spermatids. So they are also haploid. They'll produce 4 equal spermatids. As you can see, 1, 2, 3 and 4. These will also have 23 chromosomes, right? Because it was meiotic division. So they are haploid in nature. Next, the spermatids that are there. How many spermatids got formed? 4 haploid spermatids, right? So the spermatids are transformed into spermatozoa. Spermatozoa means sperms. So both are one and the same. You can either call them as sperms or you can call them as spermatozoa. So that particular process wherein the spermatids undergoes meiotic division and differentiates itself to form sperms or spermatozoa is called as spermiogenesis. So what is spermiogenesis? Now there is a difference between spermatogenesis and spermiogenesis. You need to keep in mind. Spermatogenesis, the production of the sperms by the testis is called as the spermatogenesis. What is spermiogenesis? Spermiogenesis is a process wherein the spermatids that were formed will again undergo meiotic division to produce haploid sperms. That is called as spermiogenesis. So here, because generation of sperms are taking place here. So after spermiogenesis, what happens? Now these sperms that are formed will also be, I'll show you here, these sperms that are formed. So there were 4 spermatids, right? So those four spermatids, they will undergo differentiation. Now here there is no meiosis occurring. Why no division? Because the division of the cells already had occurred, wherein they formed into spermatids. Now those spermatids will no longer divide, but what will happen? Those cells will differentiate so that they will start to look like the appearance of a sperm. So therefore 4 spermatids are formed, and how many chromosomes each spermatid will have? Again 23. Therefore being haploid in nature. Now these are the sperms which are haploid and ready to enter into the female reproductive tract and fertilize the ovum. So that is how spermatogenesis occurs. So after spermiogenesis, so now we understood that the spermatids, they differentiated to form haploid sperms, right? So after spermiogenesis, and that particular process is called as spermiogenesis. So after spermiogenesis, the sperm heads, they become embedded in the Sertoli cells and are finally released from the seminiferous tubules. So they were produced inside the seminiferous tubules, right? That is inside the compartments they were produced. So from there, they get embedded in the Sertoli cells, and from the Sertoli cells, they get released from the seminiferous tubule. So that is how they get released from the seminiferous tubule. And the spermatogenesis starts at the age of puberty. So I told you about that earlier itself, because of its significant increase in the secretion of gonadotrophin releasing hormone. Because it is at puberty, a particular hormone gets released in male, that is the secretion of a particular hormone which is called as gonadotrophin releasing hormone usually starts at puberty, and that gonadotrophin releasing hormone is very, very important for the secretion of the sperms or the formation of the sperms. That is why spermatogenesis or spermiogenesis occurs in males during the time of puberty. So this was about spermatogenesis. Now we know that all this process takes place in the puberty. So in puberty, what happens? The release of the hormone or the secretion of the hormone called as GnRH, that is gonadotrophin releasing hormone, will start. So now the increased levels of gonadotrophin releasing hormone, it will act on the pituitary gland, and pituitary gland we have seen yesterday while studying about testes, where the pituitary gland is located. So that gonadotrophin hormone that is released at the time of puberty, it will go and act itself upon the pituitary gland, and it will stimulate the pituitary gland to secrete two gonadotrophin hormones, which is called as luteinizing hormone and follicle stimulating hormone. So therefore, why is gonadotrophin hormone important? Because at puberty, when gonadotrophin hormone gets produced, then only the pituitary gland will get activated. Only if the pituitary gland is activated, the luteinizing hormone and the follicle stimulating hormone will get secreted. These two hormones are very important because they have their own specific function in the male reproductive system. What are those functions? We shall look into. So, the luteinizing hormone, it acts on the Leydig cells. So, Leydig cells we have seen, go back to the previous session and have a look. We have studied about the Leydig cells and all that there. So, it will act on the Leydig cell and it will stimulate the synthesis and secretion of androgens. Androgen is the male hormone which is very, very important for developing all the male sexual characters. If it has to develop properly, then androgen, or the male characters, if it has to develop properly, androgens are very, very important. Therefore, what did we understand now? Each of these are interconnected to one another. The gonadotrophin got released at puberty. That gonadotrophin, it stimulated the pituitary gland to release the LH and the FSH. The LH, when the luteinizing hormone, once it gets released, it will act on the Leydig cells and it will stimulate the Leydig cells to synthesize the androgen hormone. So, that is why gonadotrophin releasing hormone is very important here. And the androgen, again, it is a continuous process. Once the androgen is produced, the androgen will stimulate the process of spermatogenesis. So, therefore, if spermatogenesis has to occur, if the testis has to produce proper amount of sperms, then once the male reaches puberty, the gonadotrophin hormone should be released at correct level because every process starts from the gonadotrophin hormone. So, that is why. Now, there is one more hormone that is released from the pituitary gland. What was that? The follicle stimulating hormone, right? So, the follicle stimulating hormone, it acts on the Sertoli cells and it will stimulate the secretion of some factors which help in the process of spermiogenesis. Now, you know, right, what is spermatogenesis? The production of sperm by the testis is called as spermatogenesis. What is spermiogenesis? The spermatids undergoes differentiation to produce spermatozoa or sperms in the seminiferous tubules, that is called as spermiogenesis. So, if this particular differentiation process has to occur where the spermatids should get converted into proper sperms, then the follicular stimulating hormone should get secreted. So, that is why the secretion of the gonadotrophin at the time of puberty is very, very important in the case of male reproductive system. So, that is how the process occurs. So, same thing here, they have shown it in a diagrammatic weave wherein they have shown one large seminiferous tubule, that is the compartment. In that they have shown the spermatogonium. So, what is a spermatogonium, or it is called as the sperm germ cell, right? The germ cell, and we have the Sertoli cells that are present that get converted into primary spermatocyte. How they will undergo? First, they will undergo, that is the cells present in the seminiferous tubules, they have to multiply. So, to multiply, first they will undergo mitosis division. So, I will write here mitosis. Same thing what we had seen in the chart, right? That is shown in the diagrammatic form. They will undergo mitosis. Now, primary spermatocyte will be formed. These primary spermatocyte, later on it will undergo meiosis to form secondary spermatocyte. That secondary spermatocyte will undergo again meiosis to form the spermatids. Those spermatids will undergo differentiation to form the sperms, right? So, or here there is no need of meiosis, directly the secondary spermatocyte will undergo differentiation to form the spermatid, and the spermatid forms the spermatozoa, which is the mature sperm. So, that is how exactly the formation of the sperms occur inside the seminiferous tubules or the testis in general. So, this whole process is called as spermatogenesis. In spermatogenesis, there is a step which is called as spermiogenesis. So this was about spermatogenesis. Now since we know this process of spermatogenesis, let us move into studying the structure of the sperm. This particular topic is very, very important because it keeps repeating as a question in the exam. For either 3 marks, they will just ask you to draw the structure of the sperm, or for 5 marks, they'll ask you to draw and explain the structure of the sperm. So this is also very, very important. So in this chapter also, for sure, there are questions that arise which will fetch you good marks. So structure of a sperm. So sperm is microscopic in nature, right? Because it cannot be seen by the naked eye. So they are microscopic structure, and they are mainly made up or divided into 4 pieces, or that is, they're divided into a head, a neck, a middle piece and a tail. So, and when you look at the sperm, so if you can see here, it is mainly divided into a head, then a neck, a middle piece and then a tail. And the entire sperm is covered by a membrane which is called as the plasma membrane. A plasma membrane envelopes or covers the whole body of the sperm. So that also I've told you about. And now moving on to each of the part. What is there exactly in that? So starting from the top portion, let us study what is there in the head of the sperm. So the sperm head contains an elongated haploid nucleus. So if you see, you can see a elongated haploid nucleus that is present, which contains DNA, that is the chromosomal material. So they are haploid. Of course, the nucleus is haploid because the sperm is a haploid. And the anterior portion, that is the tip of the head, if you can see, it is covered by a cap-like structure. Can you see here? It looks just like an arrow, right? It is covered by a cap-like structure and it is called as acrosome. So the cap-like structure which is called as acrosome. And the acrosome, what is there inside the acrosome? Inside the acrosome, there are present enzymes, and these enzymes are very, very important if the sperm has to fertilize the ovum. So if it has to go and attach to the ovum, this particular acrosome is important. Why? Because the enzymes are present inside the acrosome, and that enzyme will help to detect the ovum or the egg and go and fertilize it. So it is very important for fertilization of the ovum. So that is the function of the acrosome. It helps the sperm to fertilize the ovum or the egg of the female. Next, moving on to the next part, that is the middle piece. So middle piece possesses numerous mitochondria. So they contain lot of mitochondria. Why they contain lot of mitochondria is because we all know that mitochondria is one such cell organelle which is called as the powerhouse of the cell, right? Because they supply enough amount of energy. So here the tail of the sperm requires enough amount of energy so that it keeps moving like this, like a whiplash, so that it can help the sperm to swim very fast. So that energy that is needed for the tail is supplied by the mitochondria. So that mitochondria, where it is present in the middle piece of the sperm. And this mitochondria produces energy for the movement of the tail, and this movement of the tail actually helps in sperm motility. That is, it helps the sperm to move very fast so that it can travel through the uterus, travels through the vagina, the cervical canal, through the uterus, into the fallopian tube, reach the the fimbriae region, and there fertilize the ovum that is there. So for that, it is very, very important for fertilization. For the sperm to travel, it is very, very important. So that is about the tail. So tail helps in motility of the sperm. So this is the structure of the sperm, and it is very, very important. So next, moving further, let us study about in spermiogenesis, how much amount of sperm is liberated, and all that details we shall look into. So the human male, actually the adult human male, ejaculates about 200 to 300 million sperms. Do you think all those 200 to 300 million sperms will go and fertilize the egg? No. Why? because the female will produce only one mature egg, right? So that is why. So for one mature egg, one sperm is enough. So that is why all the 200 to 300 billion sperms will not go and fertilize. They will start to compete. So which one wins the race, that will go and fertilize there. So the human male ejaculates about 200 to 300 million sperms during coitus, of which and that is actually needed for normal fertility, and at least 60% of the spores must have normal shape and size, and at least 40% of them must show vigorous motility. So out of the 200 to 300 million sperms that are produced during coitus, at least 60 sperms should have proper motility or should have proper shape. So it should have proper shape and size. But if they have, is it enough if they have proper shape and size? No, right? They should have good movement capability also, right? So therefore, out of the 60, 40 have proper motility rate is also fine. So that at least from that 40, whichever is very fast and moving, it's like a race. Will all the runners get a first place? No. Only though everybody are very quick in running, there will be only one winner. Just like that here also, only one sperm that is very quick and energetic will fertilize the ovum out of the 40% of the sperms that are there. And the sperms released from the seminiferous tubules are transported by the accessory ducts. And secretion of, and there are some accessory ducts that are, or accessory organs present in the male reproductive structure, that is the epididymis, the vas deferens, then the seminal vesicle, the prostate gland and all that, and they secrete certain substances, enzymes or hormones, and the secretions of these are very, very important for the maturation and for the movement of the sperms or for the motility of the sperms. So therefore, it is a network of process. As I told you, how the pituitary gland, how they secrete certain hormones and how they help in spermiogenesis. All that we had studied in the previous slide of this particular topic. So this is about spermiogenesis. And the seminal plasma, along with the sperm, it constitutes the semen. So seminal plasma is a sticky fluid like matrix, in that all the sperms will be present, and the function of the male accessory ducts and glands are maintained by the testicular hormones, that is androgens. So testicular hormone, it is called testicular hormone because it is secreted by the testis. How the androgen gets secreted? To brush up, I am telling you once again, during puberty the gonadotrophin hormone gets released. That gonadotrophin hormone acts on the prostate gland. The prostate gland, once it gets activated, it will secrete the luteinizing hormone and the follicular stimulating hormone. The luteinizing hormone will act on the Leydig cells, and those Leydig cells will secrete the androgen. So that is why the male accessory ducts and glands are helpful in secreting androgen, and their function is also maintained by the testicular hormone, the androgen. So this is about spermiogenesis. So the next one is oogenesis, which occurs in the female reproductive structure. Again here, oogenesis means the formation of the ovum or the egg. So the process of formation of a mature female gamete is called as oogenesis, and oogenesis usually starts during the embryonic development stage. That is, if the fetus is a female inside the mother's womb, that is during the embryonic stage itself, the formation of the, or the process of oogenesis or the production of the ovum will start. And during this time, a couple of million gamete mother cells are formed within each fetal ovary. So gamete mother cells are formed, like how gamete cells were formed in the case of male reproductive system. Likewise, here also the gamete cells get formed, and no more oogonia are formed and added after the birth. So therefore, whatever oogonia or gamete mother cells has to be formed, everything should take place in the embryonic or the fetal stage itself. If the child is a girl, later on after birth, no particular gamete mother cell or oogonia will develop in the female child. So this is about oogenesis in general. So next, let's start about the actual process of oogenesis. What happens? So first, let us look into in the form of a diagrammatic, a schematic representation. So oogenesis. So in oogenesis, these cells, that is the gamete mother cells that are there, they first undergo division and they enter into prophase 1. So they undergo prophase 1 of meiotic division and get temporarily arrested at this stage called as the primary oocyte. So here first what happens is, here also first it is mitotic division. The oogonia that has been formed. When did the oogonia form? You have to remember here the oogonia doesn't get formed at the puberty or something, like how in males it had occurred. But here, right in the embryonic stage itself, the oogonia would have been formed. So the oogonia which is formed, first it will undergo mitosis and it will produce, wherein it will have 46 chromosome producing the primary oocyte. This primary oocyte that is there, later on the cell division will start and it will enter into the prophase 1 of the meiotic division, and in that particular stage it will get arrested. It will stop at the primary oocyte. So why it will stop at the primary oocyte is because this completely it is the fetal life. That is, inside the mother's womb itself, all this process will be occurring in the baby. So that is why. Only after the birth, what happens? The next step will occur. So that is the thing. Now, don't get confused. In males, everything will start after birth, and once the male reaches puberty, then only it will start. But in the case of female, what happens? First half of the process of oogenesis would have taken place in the fetal life when the baby is inside the mother's womb. Later on, next step actually, like until the formation of primary oocyte, it will take place in the mother's womb and it will get arrested. The process will stop. Once the child is given birth and then she reaches puberty, then only the next step of division will start. So until then it gets stopped and it gets arrested. So each primary oocyte, then it gets surrounded by a layer of granulosa cells and then it forms the primary follicle. Now, a large number of primary follicle gets formed in the ovary. So, a large number of these primary follicles that are formed, they degenerate during the phase from birth to puberty, and therefore, at puberty, only 60,000 to 80,000 primary follicles are left in each ovary. So, what happens when it is in the fetal stage or in the embryonic stage inside the mother's womb? The oogonia, which are the gamete mother cells, they undergo mitosis division and differentiation to form the primary oocyte. This primary oocyte will get arrested until the birth of the child occurs. Once the birth of the child occurs and she reaches puberty, by that time what happens is a large number of follicles would have got produced in the ovary. Now, out of those large number of follicles, not all follicles will participate in the process of production of the ovula or the ovum. Here what happens is, some of the follicles that are there, which are called as the primary follicles, they actually degenerate. And only how many are left? Only 60,000 to 80,000 are left in each ovary when the female reaches puberty. Now, these primary follicles, they get surrounded by more layers of granulosa cells. So, again they get surrounded by more layers. How many? The 60,000 to 80,000 primary follicles that were there during puberty, those again will get surrounded by the granulosa cells and they form a new theca, and now they are called as, or new layer or covering, and they are called as the secondary follicles. Now, these primary follicles, what will happen? They'll when the child reaches puberty, when the female child reaches puberty, those millions of follicles that were formed, few of them will degenerate. Only 60 to 80,000 follicles will be left. Those 60 to 80,000 primary follicles that are left, they'll again get covered by granulosa cells and they will form a new theca, and now they are called as the secondary oocyte. So, this secondary oocyte and all that, they have 23 chromosomes. Why? Because After the mitosis division, it is always the meiosis or the reproduction division occurring here. The secondary follicle that is formed will soon transform into a tertiary follicle, and inside this tertiary follicle there is presence of a fluid filled cavity called as antrum. Now the theca layer that is formed, theca layer is also there in the secondary follicle. Why? Because in the primary follicle itself, the theca layer started to form, and that primary follicle got converted into secondary follicle. That secondary follicle will undergo differentiation to form a tertiary follicle. In that tertiary follicle, it is having a cavity which is called as antrum. Now, the theca layer that is surrounding the secondary follicle, it will get organized into two thecas or two layers which are called as inner theca interna and outer theca externa. And at this stage, the primary oocyte within the tertiary follicle grows in size and it completes its first meiotic division. So that is how the first meiosis will get completed. So as you can see, the primary follicle, it gets surrounded by a theca or a layer, forming the secondary follicle. That secondary follicle will again get covered by, or that secondary follicle will again get covered by more number of theca layers, and they'll form the tertiary follicle. That tertiary follicle, again it will, that tertiary follicle has a characteristic feature wherein it was filled with a fluid called as antrum. That tertiary follicle is having extra theca layers, right? Those theca layers are called as, or differentiated into or organized into, inner theca interna and outer theca externa. And at this stage, the primary oocyte within the tertiary follicle, they grow in size and they complete their first meiotic division. So that is when the primary oocyte completes its first meiotic division. First meiotic division which gets completed before the ovulation process. Because if ovulation has to occur, then the Graafian follicle has to become matured, and then it has to burst open and release the mature ovum. So that before the process of ovulation gets completed, the first meiotic division would have been completed by the primary oocyte, moving on into the secondary oocyte. So because it is an unequal division that is occurring, resulting in the formation of a large haploid secondary oocyte and tiny first polar body. As you can see here, you can see they form the large secondary oocyte, one large secondary oocyte, and they will form one small first polar body. Next, this secondary oocyte, moving further, the secondary oocyte, again it will retain bulk of nutrient, and it has cytoplasm in it which is rich in nutrient. And the secondary oocyte, it will form a new membrane. Again, it will get covered by a new membrane which is called as zona pellucida. And now it is called as the Graafian follicle. Now this Graafian follicle that is there, it will rupture, it will release the secondary oocyte or the ovum from the ovary by a process called as ovulation. So therefore what happens here? Primary oocyte gets formed, and that primary oocyte will undergo meiosis or reduction division and differentiation to form the secondary oocyte. That secondary oocyte, once it matures, the Graafian follicle will rupture, the secondary oocyte gets released outside. That secondary oocyte later on gets differentiated into the ovum, which is nothing but, on this particular secondary oocyte itself is called as the ovum, and they get released from the ovary, and that particular release of the secondary oocyte or the release of the ovum from the ovary is called as ovulation. So this is about oogenesis process. So in the exam, they usually ask the question, explain with a neat schematic diagram the spermatogenesis process, or explain with a neat schematic diagram the oogenesis process. So these two are very important. Anyone will usually come out of it. So this was about this particular topic. So in the next coming session, we shall discuss about menstrual cycle, and we shall understand about fertilization and implantation. Now we understood the gametogenesis, wherein the male gamete has been produced and the female gamete has been produced. Now how will they fertilize? How will they get implanted in the uterus? All that, or embedded in the uterus, all that we shall study in the coming session. So we shall meet again in the coming session. Thank you.